Good afternoon, everyone, and welcome to today's PHO Rounds presentation on novel aesthetic treatments, exosomes, what we know so far. My name is Anne Augustine, and I am the team lead for the Outbreak Response and Support in Communicable Disease Control at PHO, and I have the pleasure of moderating today's session. Before we start our session, I would like to provide a land acknowledgement. We acknowledge and respect the lands, sky, and waters of Ontario, including their contribution and sharing to support all life within. We acknowledge and respect the treaty, unceded, and traditional territories of all First Nations across these lands and waters, and we acknowledge and respect the traditional territories and communities of the Métis Nation of Ontario. And we acknowledge and respect Inuit connection and contribution across these lands and waters. May we respect and honor the many connections, relationships, and life journeys of Indigenous people and their indigeneity to the lands, sky, waters across all Ontario since time immemorial to this very day. May their wisdom always guide our own paths forward with open hands, open hearts, and open minds for the mutual success and benefit of all in Ontario. Thank you. Now it's my pleasure to introduce the speakers for today's presentation. We have David Riding, Dr. Maureen Savidino, Cecilia Alterman, and Brian Beck. David Riding is an IPAC specialist on PHO's Outbreak Response and Support team and a subject matter lead for personal service settings. He joined PHO in 2013 as an infection control consultant, and previous to that, he worked as a public health inspector at the Hastings and Prince Edward Health Unit. He is certified in infection prevention and control and has held his CIC since 2009. He also holds a master's in public health from Queen's University. Dr. Savidino is a family and occupational health physician with certifications from the Canadian Board of Occupational Medicine and the Certification Board of Infection Control and Epidemiology. She holds a diploma in occupational health and safety and supports IPAC at PHO. She also works in an acute care facility as an occupational health physician. Cecilia Alterman is a certified public health inspector with over 25 years experience at Toronto Public Health and is currently the manager of infectious disease infection control. She holds a master's in adult education and has taught IPAC at the Metropolitan University. So Cecilia continues to mentor public health students and has led several high-profile IPAC lapse investigations. And finally, Brian Beck is a senior advisor with the Ministry of Health's Office of the Chief Medical Officer of Health IPAC unit. He is also a certified public health inspector since 2010 and has had his certification in infection control since 2023. He has worked in many IPAC lapse investigations, including in childcare, congregate settings, and personal service settings, and he joined the OCOMO in 2024 after serving as an IPAC specialist with Hamilton, Niagara, Haldimand, Norfolk, Brant IPAC Hub. Okay, so now over to you, David.
Thank you for the introduction, Anne. It's a real pleasure to be here today to discuss exosomes, a novel aesthetic treatment that's really gaining popularity. So here's PHO's disclaimer. It speaks to the presentation being published on our website, as well as information regarding the application and use of the information. To start, I'd like to indicate that neither Dr. Savidino nor myself have any conflicts of interest to disclose. Now moving into our learning objectives. So with regards to the learning objectives, the aim is that by the end of the session, participants will be able to determine what exosomes are, how they're used, and how they're derived, identify some of the challenges and concerns with the use of exosomes in personal service settings, medi-spas, and other clinical settings, to describe the current regulatory landscape in Canada related to licensing, marketing, and use of exosomes, and finally, to identify key considerations and referral pathways when encountering exosomes in the field. So to achieve our learning objectives, we've structured the presentation as follows. We'll begin with an overview and background of exosomes and then discuss issues, concerns, and the unknowns with exosome use. Following that, we'll hear from a public health unit and their experience with exosomes in the field, as well as a regulatory overview from our ministry colleagues. And finally, we'll wrap up with a question and answer period. So to start, let's answer the question, what are exosomes? Well, exosomes are a form of extracellular vesicle secreted by all cell types, meaning that they could be of human, animal, or plant origin. Exosomes are very small membrane-bound vesicles. They're nano-sized, so making them similar in size to viruses as a reference. Their size and makeup gives them the ability to cross barriers, such as the cell membrane or the blood-brain barrier, for example, and this is a really important feature. So historically thought of as carriers of cell waste, evidence now suggests that exosomes are involved in cell-to-cell communication. This is achieved by exosomes transporting complex cargo that contains a variety of biological molecules, including proteins, lipids, and nucleic acids like DNA and RNA, and transferring these molecules to other cells. So what makes this communication aspect interesting is that the specific cargo is dependent on the cells that produce the exosome, meaning that the content of exosome changes to a degree, depending on the cells that they originated from, which then in turn influences their function and outcomes. So the source really matters. And in terms of function, this transfer of molecules to other cells has the potential to influence various biological processes, such as gene expression, cell-to-cell signaling, immune response, and tissue repair. The cargo is selectively taken up by local and/or distant recipient cells, and when taken up, they influence these cellular processes. So where do exosomes come from, or where are they derived? Well, the answer is many sources. So as just mentioned, exosomes are released from many cell sources, and they can also be found in most body fluids, such as blood, saliva, urine, milk, amniotic fluids, and more. And they can be found in the culture medium of almost all cell types. While there are many sources of exosomes, for simplicity, they can be categorized as conventional sources, so those would be human and mammal, and non-conventional sources, which are non-human, non-mammal. So most of the exosome production and isolation techniques focus on the use of human and mammalian sources, such as cells, tissues, and body fluids. And in terms of the non-conventional sources, these can be further subdivided into animal product derived exosomes. This can include honey and snake venom, bacterial, fungal, and parasitic exosomes, as well as plant-derived exosomes from sources such as fruits, seeds, pollens, and things like that. So it begs the question, I guess, are we actually talking about snake oil? Well, we literally may be talking about snake oil this time. And so why are exosomes of interest? It's because they actually show promise for several therapeutic applications, for disease diagnosis, as well as cosmetic applications, but we still have much to learn. They're being studied for their role in cancer and other diseases, and due to their natural origin and biocompatibility, they're being explored as a potential vehicle for drug delivery. And this is possible because they can be engineered to deliver that diverse therapeutic payload and directed to specific targets. In addition, they're actually showing promise as well as being part of novel vaccine delivery mechanisms. So in addition to their therapeutic potential, exosomes have the potential to aid in disease diagnosis and determining the prognosis of patients with cancer and other diseases. And this is because they've been reported in all biological fluids, as mentioned, and the composition of this complex cargo of exosomes is readily accessible via sampling and testing of these fluids. And most applicable to us today, exosomes are being introduced into the aesthetics industry through cosmetic applications. The utilization of exosomes in topical creams, serums, and masks for example, have increased due to their purported wide-ranging effects. Cosmetic applications typically include skin rejuvenation, scar and wound healing, and hair growth. In particular, exosomes have gathered significant attention related to skincare due to their rich composition of these proteins, lipids, and various molecules, which are advertised to foster healing, hydration, and skin protection. So these components are thought to contribute to enhancing collagen production, mitigating inflammation, and safeguarding the skin against environmental stressors like photoaging. And so while there's a lot of promise for the use of exosomes for a variety of applications, a key element to note, and one of the reasons why we're presenting today, is that in Canada, exosomes are considered a drug, and as such, need Health Canada approval for use. However, currently there are no exosomes of any kind delivered by any route that are approved for use in Canada. So despite this, we're learning that exosomes are regularly being used in personal service settings and medi-spas in Ontario, and it's quite possible that operators, including regulated health professionals, are unaware that exosomes have not been approved for use in Canada. So one of the complicating factors is that they're advertised online for purchase, making them widely available and accessible to operators. So focusing now on the cosmetic application of exosomes, we'll briefly look at how they're prepared and administered for this purpose. So we've sorted these into three categories. First, are exosome products that are topically administered without any disruption to the skin barrier. These would include face masks, serums, and creams that are simply applied to the skin of a client and rubbed or massaged in, for example. The second category of exosome products that are again topically administered, but this time it's in association with disruption of the skin barrier. In this case, creams and serums are rubbed into the skin following a procedure such as microneedling, laser or radiofrequency resurfacing, or dermabrasion, for example, all of which create micropores in the skin and function as a portal of entry for the exosome product in order to penetrate further into the client's skin. And finally, exosomes can also be delivered through intradermal, subcutaneous, or intravenous routes. So I will note, though, that to date, PHO has supported health units that are dealing with these topically administered products only in these Ontario premises. So at this point, I'll now hand over the presentation to Dr. Cividino to discuss some of the issues, concerns, and unknowns with exosome use.
Thanks, Dave. I'm going to discuss some of the safety issues and adverse outcomes, including infection risk that is inherent really with the design and function of exosomes, as well as talk about some of the manufacturing process issues and ultimately issues that are just due to the application of the exosomes themselves. Exosomes can either accelerate or inhibit the process of infection, and in both cases, exosomes make possible connections between host cells or between pathogens and host cells. Exosomes containing proteins and RNAs derived from pathogens can promote infections in three ways. First, by mediating further infection by transmission of pathogen-related molecules. Second, by participating in the immune escape of pathogens. And thirdly, by inhibiting immune responses by favoring immune cell apoptosis, which is the programmed natural death for cells. On the other hand, exosomes can play anti-infectant roles, first by inhibiting pathogen proliferation and infection directly, and second by inducing immune responses, such as those related to the function of monocyte macrophages, natural killer cells, B cells, and T cells. Exosomes have been described as acting like a bridge, where molecules related to pathogen infection can really go in either direction and can either then promote infection or inhibit infection. An example of immune escape is the release of hepatitis A virus after being anchored within the host cell membrane, protecting the virion from neutralizing antibodies. Therefore, the exosomal route can avoid contact between the virus and neutralizing antibodies, allowing further infection spread. Exosome from virus-infected cells can also spread microRNAs of non-host origin, making them undetectable by the host immune system. Some recent studies have reported hepatic exosomes can help transmit hepatitis C virus infection in vitro, and again, by HCV being potentially protected from antibody neutralization. This has also happened with hepatitis E virus, where in fact particles have been found in an exosome fraction, and these have continued to be infectious and were unable to be shut down by neutralizing antibodies. The mechanism of action for that is still actually being explored. There are also many manufacturing process issues for handling and administration errors that can occur and can be implicated in infection transmission. We've seen examples of HIV transmission through platelet-rich plasma administration during vampire facials, which is trademarked apparently, and where the client's own blood then is centrifuged on site and the platelet-enriched plasma is applied to the face following microneedling and microneedling with radiofrequency. In 2018, the New Mexico Department of Health investigated a case of HIV with no known risk factors. This was following a PRP procedure in an unlicensed spa. 4 other spa clients and an additional sexual partner of a spa client over the next weeks to months were also diagnosed with HIV. They were genetically linked and all related to poor IPAC practices. This is just a great graphic that really illustrates what we've been talking about. So regardless of which microorganism, so you can see whether it's virus, bacteria, fungus, or parasite, the infection then into the cell secretes the exosomes, and the exosomes then can either carry entire pathogens, pathogenic genes, or pathogenic proteins, and the impact on the target cells then being either direct infection, alteration of the gene expression in the nucleus, or the influence of the pathogen virulence factors. So looking at challenges with producing exosomes, a key challenge is really where they're sourced from. Exosomes sourced from human sources are more likely, understandably, to contain human pathogens, but that doesn't mean that exosomes sourced from non-human sources are safe. Exosomes sourced from animal sources can pose a risk of transmitting zoonotic disease, with an example such as Q fever if proper controls are not in place. A key component of exosome manufacture is a thorough screening of exosome donors, and the source needs to be screened for bloodborne infections, including hepatitis B, C, HIV, as well as for bacterial pathogens. Manufacturing techniques are important based on their nanovesicular size, makes purification of exosomes particularly challenging, and breaches in aseptic technique can also introduce contamination. There's currently also no agreed upon process on terms of how best to purify exosomes. Finally, quality control is both key to ensure the product contains the desired levels of exosomes and also to ensure that the product is free of contaminants. Since 2020, the US FDA has issued several warning letters related to the manufacture and distribution of exosomes. This includes 4 inspections conducted across 3 facilities that were manufacturing exosomes without regulatory approval. They identified 50 infractions across 25 different offenses. Failure to test donors for certain pathogens like Zika virus was one, poor quality control, failure to screen donors for other tests, poor aseptic technique, unlabeled product, which is found in quarantine freezers, failure to test products before they were released for use, failure to reject batches after donor plate showed microbial growth, distribution of products after testing positive for gram-positive cocci, and distribution of product after the donor blood tested positive for hepatitis B. There's a list of additional sterility failures with products contaminated with a number of pathogens, as you can see here: gram-positive cocci, gram-negative rods, acid oryx temperans, Clostridium perfringens, which should have a second R in there, Enterococcus faecalis, E. coli, Klebsiella pneumoniae, Cocuria varians, strep species, and hepatitis B virus. There was one instance where a patient later tested positive for E. coli after an initial sterility failure was not investigated. Other IPAC issues include product containers for sterile products that were being stored in open bins with lids that had been bought in a bulk store, and there have been no disinfection or sterilization of these prior to use. As well, unhygienic practices for sterile preparation, where hair was not covered, skin was not covered, and there were no use of sterilized gowns, gloves, bouffant caps, or shoe covers. Also breaches in aseptic technique, including processing of umbilical cord blood without sterile gloves. In Nebraska in 2019, there was an outbreak of 5 E. coli sepsis cases, all occurring within hours of administration of intravenous exosomes. It was through the quick action of Dr. Maureen Tierney, who led the Nebraska Healthcare Associated Infection and Antibiotic Resistance Team and shut down the unlicensed facility that helped prevent more infections. She received the inaugural McKnight Prize for Healthcare Outbreak Heroes in 2020 for her investigative efforts. Those exosomes were sourced from harvested human placentas. The product was unregulated, no FDA approval, and not part of a clinical trial. The company relied on internal protocols for safe production. And within 2 days, the bacterial isolates from the contaminated product matched the clinical specimen, resulting in statewide health alert and an FDA notice. Other case reports, 8 individuals who developed erythematous and indurated papules or nodules at the site of exosome injections, onset ranging from 2 weeks to 3 months post-treatment. A 50-year-old woman who developed papules and nodules 7 weeks after receiving exosome injections with a product that had not been officially released, and she was diagnosed with foreign body granuloma. As well, skin necrosis in a 36-year-old man who received intradermal injections of an exosome product that had been mixed on site and 3 days later presented with skin necrosis. Exosomes have a short half-life and poor zeta potential. This makes them less stable as a product. Zeta potential is a measure of the electrical potential differential at the interface between the charged particle and the surrounding fluid. It's very important in terms of colloidal stability, and when it's low, it can lead to aggregation or sticking together and flocculation. If the product isn't stored properly or the dosing rather is improper, then the desired therapeutic effect won't be obtained. Factors impacting the therapeutic effect include delivery of exosomes when aggregation occurs either prior to or after delivery. As well, the immune response may be triggered more quickly, and circulation time for the exosome may be reduced. All these challenges can impact degree of positive therapeutic effect on target site. Optimal dosing of exosome treatments can be difficult to determine. Considerations include the delivery methods, inherent biological properties, such as the short half-life and zeta potential, and how parent cells impact the exosomes that they created and their subsequent therapeutic effect. Additionally, all of the improper handling concerns that we have regarding medication administration also apply to exosomes and storage issues as well. So in conclusion, exosomes offer exciting promise in cancer and neurologic disease, use as a potential vehicle for drug delivery, and there's growing interest certainly in their application in the field of aesthetics. Despite exosomes having no Health Canada approval for any type, human, animal, or plant, or approval by any route of administration, topical or injectable, they're currently widely in use in aesthetic services in Ontario. There remain significant safety considerations in the sourcing and manufacturing of exosomes, as well as in their handling, administration, and storage. And finally, as discussed, reports of serious infection and adverse health outcomes, including sepsis, with intravenous use. So that concludes my presentation. If you have any additional questions, you can reach us through this email. And at this time, then I will turn it over to Cecilia Alterman, who will speak to us about the experience on the ground in the field from the Public Health Unit. Thanks very much, and over to you, Cecilia.
Thanks, everyone. So I'm here to speak about our experience as a health unit in the field. Next slide, please. So what's interesting is that exosomes were first brought to the attention of Toronto Public Health a few years ago by one of our amazing inspectors, Shelley Wagner, who just happened to be reading about the use of exosomes in cosmetics and how these products were becoming quite popular in the US beauty industry. So she brought this to attention of the team. We had never heard of exosomes in cosmetics before. And, we wanted to be prepared in case we started to see these products in Toronto. So the first thing we did is we contacted Health Canada to learn about what is it? Does it require a drug identification number, a DIN? We wanted to understand the risks, if it could be used topically at a minimum versus with like an invasive device like microneedling. So obviously we had many questions as we knew it was a matter of time before we'd probably find it on site. Since it was during COVID, we didn't hear back from Health Canada at that time. So life went on, and it wasn't until about 2024, last year, that we noticed, the name Exosome in products being advertised as part of microneedling style services in our personal service settings. So, of course, my amazing team, as they were out there doing their inspections, we started to find the products on site. It was during this time that we reached out to the province to help us learn a little bit more about exosomes. And again, what are those risks? And as we suspected, as we started to look into these items, they had no DIN, no natural product number or NPN. And then when we searched one of the company websites for more information, we found that the manufacturer suppliers, they were claiming that their products were USA FDA approved, which we later found out, like in Canada, none are approved. We also started to find various kinds of exosome products out there, including, like human cell origin, animal, and synthetic means. Next slide, please. So this is an example of a few of the products we've seen out in the field. There are many more. In the first picture here, if you read the ingredients, which I know it's a little small, it actually states human cell exosomes. In the second one, the label indicates it's an exosome-like cosmetic regenerative complex. So it has active ingredients such as amino acid complex, peptide complex, and hyaluronic acid. And then the third one, it's pretty obvious. We've got some Australian sheep placenta. Next slide, please. As we saw these products out in the field, we just continued to refer them to Health Canada. And so we would take pictures, identifying the manufacturer, supplier, all the information we could find. And soon after, Health Canada did respond and verified that there are no products approved in the US or Canada for consumer use, as mentioned by Maureen and David. They also indicated in their response that if they were approved, they would be regulated as a drug or a biologic health product and not a cosmetic. So therefore, they would still not be permitted in personal service settings for aesthetic use. Many manufacturers and suppliers, what they were doing is they were letting their operators know, and then, of course, telling us later that the product was approved as a topical cosmetic and therefore did not require DIN or NPN. So that's why operators were obviously very shocked when we were going in, telling them that it was not an approved product, even as a topical cosmetic. But so fortunately, as we were telling operators to remove them off the shelves, we received full compliance. Next slide, please. So as mentioned earlier, we notified PHO, the Ministry of Health, to help us understand the health risks, we wanted to know also if other health units have been seeing this product, if there were any other concerns, and it appeared that it was a new discovery. With the support of the province, we wanted to also bring awareness of the risk of these products and popularity to Health Canada, at least as a start, so that perhaps there could be a nationwide alert in the future. Since we first observed exosome products on site, we have continued to give verbal orders under Section 13 of the Health Protection and Promotion Act, ordering to stop the use and removal of the product from the premises. And again, operators have complied with all orders. We are also recommending operators stop all advertisement of the product and services associated with their use. Next slide, please. So we continue to document our findings and continue to report to Health Canada and the province. And in doing this, actually one of the larger suppliers of Exosome cosmetic products actually contacted Toronto Public Health to speak to me to inform us that they've removed all exosomes from their future products. The individual sent me pictures of the new items with the ingredients, and they indicated that they were quite understanding of our actions in ordering the removal from the settings. So that was great news. So it looks like at least one large supplier is starting to, or manufacturer is starting to remove exosomes. We also posted general information for the public and operators on our BodySafe website. Next slide, please. And here you'll see a screenshot of our BodySafe main page. There's a banner at the top that has the warning. And so when you go there, you can just click on the aesthetic page, which will provide you with more information about exosomes and the risks. And of course, we're asking anyone, if they see this in a personal service settings, to please inform the program. I should say that actually this week, ironically, one of the inspectors that was out found another new exosome product. So I will be sending that over soon to, to the provincial folks. But what was also helpful is that during their interaction with the operator, they were able to refer, this individual to our website so that they could read themselves, what the risks are, and see that we are making this, an important issue and warning others. Next slide, please. So some suggested next steps is basically to do what we're doing now, continue to inform and update our partners of the risk of these products, including health units, which I'm hoping there are many on this webinar, so that eventually we remove, we educate those that have this item in their stores, remove them from the shelves, and also put some pressure on suppliers and manufacturers to remove the ingredient altogether. We're hoping also regulatory colleges are informed so they can notify members who are practicing in medi-spas or even in their own practice. Again, stop the use and stop the purchase of these products. And finally, as mentioned earlier, we would like Health Canada to issue a nationwide warning on the purchase, sale, and use of these items. So that there's nothing coming in also from within Canada as well. So that concludes my presentation. Thank you so much. I'm now going to pass it over to Brian Beck from the Ministry of Health, who will continue with the final portion.
Thank you, Cecilia, for sharing some of your experiences with Toronto Public Health and kind of the details to date as it relates to exosomes. So just to wrap things up, for today's content for the presentation, I'm just going to spend some time looking at the regulatory landscape for exosomes themselves. Just a couple quick objectives, and I'll try to make my way through this relatively quickly so we can spend some time with the Q&A towards the end. So my portion of this presentation, we're just going to kind of review some of the regulatory landscape in Canada as it relates to licensing, marketing, and use of exosome products. We're going to identify some key considerations for the different stakeholders that may be a part of this call, whether that be public health unit level staff or potentially regulatory colleges. And also touch on some of the referral pathways when it comes to exosomes. Okay. All right. Just got to get used to that, a little bit of slowness there on the turn. So just to start, in Canada, personal care products are regulated by Health Canada as part of the Food and Drug Act. So what the first step as part of this regulation of personal care products is to properly classify the products as either cosmetics, which are covered under the cosmetic regulations, or drugs, which would be under the food and drug regulation side of things. Definitions for both of these terms can be found within the Food and Drug Act, and they've been included here just for your awareness. If we're looking at these definitions on the face, they seem relatively straightforward, but what we often see in personal service settings or in medi-spa type of applications is that they can be quite tricky. Oftentimes, the products that we may come across in these types of settings have characteristics of both. So they have characteristics of cosmetic applications as well as drug applications in this case. So in this situation, Health Canada goes through and does a little bit of a case-by-case assessment as part of this, and they use a set criteria of the following three components. So the first thing that they're looking at would be the representation for use. So the indications of use or the claims that may be present on the product, the label, or as part of advertisements about how they would be used. The composition of the product, so the ingredients, the components, and the structure of the product, and the level of action. So the level of action one is something we're going to touch on a little bit here as well. So just generally, cosmetics must lack that precutaneous absorption and should not have to be absorbed below the skin to achieve their desired effect. So cosmetics themselves cannot be administered via ingestion and inhalation or by injection, with the lone exception for cosmetics in this case being tattoo ink, which we're all familiar with on that side of things. So at the total picture here in determining whether something is considered to be a cosmetic or a drug, we're not necessarily completely relying on the risk, but just how they're actually kind of what the end result of this review would sort of turn out here. So before we get into kind of the specifics that we've come across as part of this investigation on exosomes, just want to kind of demonstrate some of the intricacies that are involved in determining whether something would be considered a cosmetic or a drug in this particular case. So microneedling is a very common service. Those that work in public health would be very familiar with seeing this type of service within personal service settings and the various different products or serums that may be used in combination with microneedling. But as we work through here, you can kind of see how this can become complicated in trying to make a determination in the end. So very generally here, if a product is administered by microneedling, it would not be considered a cosmetic. That one's the most clear as part of this. So if it's administered as part of that microneedling process, it would be considered a drug in this case. However, the timing does impact things a little bit further as we work through. So if this product, whatever it may be in this case, not specific to exosomes quite yet, if it's applied before and not at the actual time of the administration, and the claims on the product that is being used are specific to cosmetic functions, so thinking about things like moisturizing or cleansing or conditioning of the skin, then they could potentially be a cosmetic. However, if the product itself has a therapeutic claim prior to microneedling, it would be considered out of scope to be a cosmetic process. So there's some additional details here that are included as part of this presentation that could be useful afterwards. If the substance is applied before microneedling, but will be absorbed deeper into deeper layers of the skin during the procedure, then it would be considered beyond the scope of a cosmetic. So just the basic facts of how and when the timing component of this and the contents or the intended use of the product can vary whether this would be considered a cosmetic or a drug in this particular case. So just switching gears here and kind of reviewing the sort of Health Canada or the authorization as general. We've heard kind of bits and pieces of this as part of the webinar today already, but just wanted to kind of provide a little bit more of an overview of what this actually looks like at the moment. So with drugs, there's two potential authorizations that are possible. The first one is related to those that are a part of a clinical trial. So they would receive a no objection letter in that case. And the clinical trial itself could be looked up through the database that's included here. The one that we're most familiar with and see quite a bit here would be more so the market distribution part of things. So in that case, the manufacturer would have a notice of compliance, and they would also have a dedicated drug identification number as part of this particular product. Cosmetics, on the other hand, and Cecilia talked about this a little bit here as well, go through a little bit of a different process. So in the case of cosmetics, the manufacturers in this case, or importers of the product itself, are required to let Health Canada know within 10 days of their first sale in Canada. So there is no pre-market review as part of cosmetics, but they are regulated in a post-market type of system. So what the important thing here, that they would have a sort of cosmetic notification form. And in some cases, we've seen this actually be promoted or advertised by service providers as having a Health Canada approved type product. But what isn't the case as it relates to cosmetics in this type of situation is that this notification form that they've submitted their notification, it does not constitute approval for sale by Health Canada in that case. It does not agree that the product itself is properly classified as a cosmetic and not as a drug. And the third one here is that it does not confirm that the product complies with all legislative requirements that they may need to meet as part of things. So we've heard a little bit about this already, but just wanted to reiterate where things currently stand as it relates specifically to human-derived exosomes. So all human-derived exosomes intended for administration, whether topically applied to humans, are considered drugs under the Food and Drug Act. So the rationale that we've been provided with in this particular case from Health Canada is that they are associated with inherent therapeutic potential, and they do have a biological activity associated with it, as Dr. Savidino covered earlier. Due to this, it requires authorization from the Food and Drug Regulations as biologic drugs prior to sale. As of early last week, this remains the case that there are no Health Canada approved or authorized exosome-based products for use in Canada. Neither the NOL or the NOC and the DIN in this particular case have been provided to date for exosome-based products, and therefore they cannot be legally sold or marketed in Canada at this point in time. As with anything at this type of situation, this is certainly subject to change. This is the most current information that we have available to date at the ministry side of things. So just to cover, and Dave alluded to this earlier, that outside of human-derived exosomes, there are other exosomes that are available currently on the market, whether they be plant or animal-based. There's many different options that are out there. A little bit different than human exosome-based products. These are assessed more on a case-by-case basis. So Health Canada in this situation would be looking at factors like the source, the representation, the composition, the route of administration, and the therapeutic activity that may be associated with the product. So on the right side of your screen, what you're seeing is two examples that have been brought forward as part of the investigation. These are both claims that have been made by specific manufacturers as part of their product. The first one provides very clear sort of overview that would make it a drug in this case. So they have purported claims of they have intercellular communication bridges to enhance and activate skin cells, and that would be sort of the red flag kind of signal in this case that it would be considered a drug. And that is what, in this case, that is exactly what has happened, that this product has been identified as a drug. And the same with product number 2, that is a milk-based exosome. It has less details associated with its potential, but simply based on how this product is used, that it's intended to be used and sort of puncture the skin, it would be also assessed as a drug in this case. Okay, we're going to jump around a bit. So a little bit of details of kind of where things are at the current time. So under the Food and Drug Act, Health Canada has the authority to take compliance and enforcement actions as part of any non-compliance with the Food and Drug Act or its associated regulations. They would have the ability to verify if non-compliance has occurred. They'd be able to take compliance and enforcement actions in line with their compliance and enforcement policy. The type of action really depends on the facts in each particular situation, and they are very specific to each situation in that case. So as part of this process, and as Cecilia mentioned, we've worked very much collaboratively since this has been identified over the last little while here, to bring these products that have been found in Toronto, but also in other areas within Ontario. So whether it be suppliers or personal service settings that are offering this service to their customers, we have referred these over to Health Canada for their review. And as part of that follow-up, they have informed us at this point in time that they have opened an investigation as part of this particular situation in Ontario. So really, our role has been fairly limited in this case from the ministry level, really trying to gather and bring the details forward and work more as a liaison with Health Canada to bring these to their attention. But also, as was been mentioned with Cecilia and both Dave and Dr. Cividino, we felt that it was very important to raise awareness on this particular issue as we see exosomes expanding quite readily across the province at this point in time. So very quickly, I know I'm running out of time here, and we got a couple questions, so I'll wrap things up in. Just a few things that we want to leave for considerations for our different partners that be a part of this call. So for our public health folks that may be on the line, some things to think about if you were to come across exosomes in your particular jurisdiction. Are there potential IPAC concerns associated with the products, whether the product itself or how it's being applied or utilized as part of the service? Is there a potential health hazard in place? So is this something that authorities under the HPPA may be needed to mitigate against? So use of a Section 13 order. But just to reiterate, this is really, this is based on a local decision at this point in time. And for those that may be joining from the regulatory college perspective, a few key takeaways for consideration to review as part of this. There may be opportunities to provide education to members, but on the other side, there may also be questions that come to regulatory colleges about how they may be able to use, what are the requirements, what are some things that they need to look for to make sure that they're using an appropriate and safe product in the end. So with that, I'm just going to turn things over. I've got a couple slides left here, but in the interest of time, I'll wrap things up and we can kind of head over to the Q&A portion of things.
Well, thank you so much, Dave, Dr. Cividino, Cecilia, and Brian for a very insightful presentation. We'll now move into the Q&A segment for question and answers from to answer questions from our live audience. So I'd like to welcome Catherine Kerr and Ray Gullickson, who will be joining us in the Q&A segment. Both Catherine and Ray are infection prevention and control specialists at Public Health Ontario. A quick reminder to everybody, if you haven't already, please enter your questions in the Q&A pod. So over to you, Catherine.
Thanks, Anne. Thanks, all the speakers. That was an incredible presentation. The first question, maybe Brian, I can direct this to you. I know we touched on it, but does the FDA regulate exosomes?
As of right now, the best of our knowledge here, they're in a very similar situation as that there's no approved products available for use related to exosomes. They've taken a little bit of a different stance with it, and as Dave and Dr. Savidino highlighted earlier, they have put out sort of public advisory notices, which is not something the case that we've seen yet in Canada.
Okay, thank you. Cecilia, I think I've got a question for you. Do you do any follow-up on the verbal orders issued to ensure that the operators remain in compliance?
Okay, so yes, we do. We conduct a reinspection as part of our process to ensure that the product's not there. But after that, you know, if they sneak it back in, we wouldn't know that until the next routine inspection or unless there's a complaint.
The next question is for Maureen and Dave. If not happening already, would PHO consider work towards creating practice standards in place on the manufacturing and use of exosomes?
Thanks, Ray. I'm happy to address that one, and thanks for the question. Really, like guidance for the manufacturing and the requirements for companies to follow kind of good manufacturing practices or GMP would fall under the purview of Health Canada and really would be out of scope for Public Health Ontario. So Health Canada would be the best group to target that type of response.
Okay, thanks. This is a really interesting question. Maybe Cecilia or the group can answer, but what should we be looking out for in the list of in the ingredients list of products to cue us that these might be exosomes?
So, I mean, I can only tell you a little bit of what we've seen. So maybe somebody from the province might have more information. But basically, when you see anything that says exo, exosynthetic, placenta, human, any of those type of keywords, the inspector will take a picture, notify me right away. And even if we're not sure, we'd rather be safe than sorry, and we'll send it over to Health Canada and also the province, so we could just get more information. Yeah.
So the next question is for Brian. So for drugs, there is a DIN or DIN number. Is there a way to verify that cosmetics are approved by Health Canada?
So it's not quite as easy to look them up as it would be with the drug identification number, simply because they may have kind of submitted their notification and that would sort of satisfy the requirements. But if there are concerns, we can definitely kind of assist on that front of things and kind of dig into it a little bit further.
Thanks, Brian. For Cecilia, so you showed the website information, the Body Safe. In addition to the web page, have you done any social media posts about the use of exosomes?
Yes, not yet. We haven't gotten there just yet. We might do that, but we wanted to just see if there was more information that we could gather before going to social media. But it's in the radar.
The next question is also for Cecilia. Is would you be able to share the rationale and wording used for the Section 13 orders?
Yes, I can do that.
Okay, thanks. This is a interesting question. So Benev exosomes say they are Health Canada approved for topical use, not injection, and spas are using these. Is that accurate?
Yeah, that was one of the first ones that came to our attention, actually. So in this would be the case of them claiming approval as a cosmetic. So they may have submitted their notification form to cover them off as part of the requirements for cosmetics, but there's been no actual approval as a drug, and that's what human-based exosomes are considered in this particular situation. This is exactly one of the challenges that we've run up against in this case. Is there's been more than one that have promoted that they have approvable status, but their approval is related to cosmetics, or that just in fact that they've actually submitted the notification form.
So the next question is, I guess, for whoever feels most comfortable answering it, and that is, what is being done about the off-the-shelf products sold directly to consumers, such as products from retailers like Sephora?
So I can answer that. Funny enough, I didn't know that. So because we have many Sephoras in our area. So we will look into that. What we would do is Sephora is considered a personal service settings. They do provide services within their retail store. So we would follow up as we would any other PSS if we saw the product. One of my staff actually saw face mask in a grocery store where it had exosomes as well. So it's popping up everywhere. And I think it's just, again, hoping that there's a little bit more education information out there for everyone. So all various types of retailers. Unfortunately, we don't have authority there. So she can just go tell the manager offline.
Maybe just to add to that one quickly, Catherine. As part of sort of our work with exosomes and the investigation that's ongoing with Health Canada, we have done a fairly thorough sweep in terms of any potential suppliers or products that may be out there. Now, the list is continually getting bigger, but that is something that we've tried to sort of pass on to Health Canada to demonstrate the sort of how big this issue is in Ontario and how big it's growing. So, there's capability of including additional products as part of that investigation, but we did try to do a thorough sweep of, all of the types of products that were currently available within Ontario.
Thanks for that. There's a question, they're sort of similar. I'm going to combine them. So it's, are registered nurses who provide cosmetic services allowed to provide exosome treatments? And then in follow-up, so if you see a medical clinic that do exosome injections, would you refer them to the regulatory college?
So maybe I'll take a stab with
Yeah.
this one.
Thanks, Brian.
In terms of the first one, so that was regarding registered nurses providing cosmetic services. Are they allowed to provide exosome treatments? So right now, as we've sort of touched on, there actually are no approved products. But in terms of the oversight of that regulatory health provider, that would be up to the college that they belong to in this particular case. So yes, a referral could be made to the College of Nurses, for example, expressing this concern. And that would probably be the best way to bring that individual provider situation to light.
So I'm hoping that, Maureen, you might be able to answer this next question, and it's, are human growth factors considered exosomes?
Not to my knowledge, no. And certainly just sort of backing on a little bit of what Brian was saying about from an FDA side of things, that if we're looking for any kind of clinical indications for use of exosomes, where they may get approval is in some type of clinical trial. So if we looked at earlier when Dave was talking about with cancer, neurologic diseases, vaccine delivery, some of these other things, but those would be case by case specific approvals that would happen, but not to my knowledge in terms of human growth hormone being exosomes. Hormone, certainly.
Okay, thanks everyone.
Okay, As we wrap up today, the PHO round session, I'd like to thank David, Dr. Savadino, Cecilia, and Brian for presenting. And I'd like to thank everyone who joined us for today's events and for your really interesting and thoughtful questions. It's been great. So you can expect to receive a brief and anonymous survey for today's session. Please try to complete this to help us improve our programming. And thank you for joining us and have a wonderful day.